Okay. Okay, so the research question of our final presentation was how does varying soil moisture levels affect the diversity of plant communities in the given ecosystem? And so here's the methodology. And basically, like we selected multiple sites within the San Joaquin Marsh and the Alchard Park to represent a gradient of soil moisture conditions from very wet to very dry. And then, like, you recorded the key characteristics of each site, such as the elevation, slope, vegetation type, soil moisture level, and proximately or proximity to the water bodies. And then you collected soil samples in a vial. And then the eDNA present in the soil can be used for meta barcoding. And the bioinformation is used to generate data sequences. And so the taxonomy table and metadata table help to identify plant species from given sequences. And the variables that were used was the soil moisture level and taxonomic information. So here's like the data table and analysis. So samples were collected from the Altrish Park and San Joaquin Marsh, and the ponds that it was collected from was 2, 3, and 4. And then the location data included GPS coordinates, altitude, and proximity to walking trails. And then the vegetation sampled included grasses, curly dock, honeysuckles, and California bulrush. The moisture levels range from extremely dry to moist. And then notable conditions include varying plant types and soil conditions. And then like right here on the right side is just like the data table that we use. So the research question again was how does varying soil moistures affect the diversity of plant communities in the San Joaquin Marsh and Alchard Park? So our motivation for this question was to protect and conserve the biodiversity. And then our field trip to the San Joaquin Marsh provided a firsthand look at the complex interplay between soil moisture and the plant communities. As we traversed the marsh, we observed firsthand how plant species were distributed across areas with varying levels of water saturation. So our hypothesis, like to answer this question, was that plant growth relies on the level of soil moisture as it facilitates nutrient uptake. Therefore, less plants will be able to grow in the area with a lower level of soil moisture. And like this little picture here is kind of just like showing like what we predicted the plants would look like with like the varying moisture levels, just like for a visual representation. And so here's like a chart that we had to show like our prediction for it. So like on the bottom is like the soil moisture level with it going from very dry to moist. And then the y-axis is saying the observed diversity of the number of plant species. And so like basically it's saying that as it goes from like less dry to like moist, like the more like plant species there will be, or like the more diversity there will be. And so we would expect the soil moisture level is positively correlated to the diversity of the species of plant communities in the San Joaquin Marsh and Aldridge Park. And so the conclusion that we came to was that our predictions were true, and we can predict an increase in diversity of plant species on average with an increase in soil moisture level in the San Joaquin Marsh and Aldridge Park. We can conclude the soil moisture levels is a key factor that should be focused on in protection and conservation of biodiversity. Habitats with high soil moisture levels, such as wetlands, should be kept safe to protect the biodiversity. And then some ethical considerations to make are to make sure to conduct fieldwork in a manner that does not disturb the environment and avoid polluting the environment to make sure to clean up after. And these are just some practices that like also we practiced when we went to the San Joaquin Marsh to like take those soil samples. And that's it.
And then we have to talk about it, give feedback for 2 minutes. So, what did you guys think?
I thought that it makes sense that the moisture would be positively correlated with the diversity because when there's nutrients in the soil, then with the water, then more plants will be able to grow. So there will be a positive correlation.
I agree with what you said. Basically, I was going to say the exact same thing.
I liked how I liked just like how the slides looked in general. They were really like easy to read and to the point, and I like the idea about the moisture. I didn't do that one, so it was pretty cool.
Oh yeah, I also liked how you described each like the graph, like how you describe it individually. I like that.
And also, including ethical considerations, I think that was a good slide to have. So that way, like, you know that the experiment was ethical. Yeah.
All right, and then what do we do next?
I think the next
I
person
think
could just go.
Do I just
I
go
think we
next?
can, yeah, do you guys want to do the feedback slides like after everyone goes or while each person goes?
I'm not sure. Up to you guys.
We can do it now if not.
Okay. Wait, does everyone have the link for those slides?
Do you mean for like your slides or as in like what we have to write the feedback?
Like the feedback slides.
Yeah, it should it should be I can send it to you if not. I can share it.
Because we're on one right now, so I don't know if you had that one or if you made like a separate one.
If not, can you send it to me?
Yeah.
Okay, thank you.
I sent it on like text.
Let me know when you guys are done doing the feedback.
I'm done.
I'm done.
I'm also done.
Okay. Can you guys see my screen?
Yeah, we can see it.
Okay. All right, so I'm Matthew Dyer, and I'm from Team 14, Lab 10. And our question was, how does the distance from the walking path affect abundance of animal eDNA? So we did this in the San Joaquin Marsh, and the San Joaquin Marsh is a preserved wetlands found in Orange County. It is the remains of the once extensive wetland mosaic along the coast of the freshwater marshes, and it's part of the University of California's Natural Reserve System, NRS, which is the largest university-owned network of natural reserves in the world. So what they do is they buy a lot of like open land that companies were going to buy just to build corporate housing, kind of like you can see in the background of the left picture. The land was going to get turned into just buildings and parking lots, but they buy it just to keep the land how it is for the animals and all the other organisms. And what is in the marsh? There are 600 different confirmed species who live in the marsh, and that includes ducks, turtles, raccoons, coyotes, fish, plants, and more. And the habitats range from marshlands, shallow ponds, channels, and dry upland habitats spanning across 300 acres. And for our experiments in here, there's different walking paths, and then there's different types of pods, which are for mainly for research. And then the question is, how does the distance from the walking path on the marsh affect the abundance of animal eDNA? Our reasoning is to investigate the effects that human intervention, creating walking paths, etc., have on wildlife and biodiversity, and how the presence of more vegetation affects the presence of wildlife and biodiversity. So pretty much like how does human intervention change how the abundance of like animals and sightings and stuff like that. And right here with the graph, with our hypothesis, as the distance from the walking path increases, the amount of biodiversity in the eDNA will also increase. So we have on the x-axis the distance from walking path in meters, yeah, and then the y-axis the amount of eDNA. So as the distance from the walking path increases, we expect also an increase in amount of eDNA. So we can predict that since there is an increase in vegetation as we proceed towards the pond and away from the walking path, this increase in vegetation will also correlate to an increase in biodiversity. Um, and our data was collected from different areas in the pond and samples taken from different sediments containing eDNA. And eDNA is processed and sequenced in metabarcoding. These sequences can be translated using bioinformatic techniques such as PITS or PITS, which analyze plant sequences in order to determine which species are present in the sample. The expected results of these samples will vary depending on the distances and environments that the samples were taken from. And here's part of the metadata table. A metadata table is a table that contains information about entities in a local data model. So we have from our data collected, we have our longitude, longitude, altitude, how precise we were. We have photos of the vegetation, distance from walking path, the type of soil, and then the IDs from plants sampled from, habitat, and moisture level. And then in our taxonomy table, this is just a little screenshot from it. There's a lot more, but the science of naming and describing the organisms, putting them into a class is taxonomy. This includes all plants, animals, and microorganisms. Um, there's all sorts of classes, but we're more focused on the family, genus, and species, which you can see is right here. And then with our data, after reviewing the metadata table, the soil further away from the path tend to be more moist, as opposed to the very dry soil near the path, and the moist soil is more conductive towards plant life. If our predictions about the increase in biodiversity in plants with a higher concentration of vegetation are correct, this will also result in an increase in the abundance and/or diversity of eDNA. And vegetation and water abundance in ecosystems are an important part of fostering biodiversity. The distribution of biodiversity measured here can have implications on human interactions with ecosystems, such as plants, such as the paths constructed on the preserve. And that's about it. Thank you.
I liked how uh put a lot of images to like describe what you were saying. It wasn't just all text. There was like more images and text, like you divided it.
I liked how you like thoroughly described like everything, like even like what the San Joaquin March is, just to give like some like background, like information to like help like someone who doesn't know what it is, like know what like this whole process was.
Yeah, I was gonna say I liked your introduction, like how you explained everything and like showed like different diagrams and pictures. So it's more easy to envision for someone who doesn't already know what the San Joaquin marshes are.
Charlotte, you can go next if you want. I can go last.
Okay, I'll just finish writing the notes on his slide real
Uh,
quick. And okay, is everyone ready for me to present? Is that okay? Okay, cool. Let me figure out how to share my screen. Okay, actually, let me not do that. Okay, I'll start. So, my presentation. Okay, second actually. Okay. So my presentation, my research question was, what is the correlation between distance from walking path to species diversity present at each transect? So it's similar to the last presentation. The motivation behind our question is finding the relationship between different locations and species. So we're trying to find the diversity and seeing if the location affects anything with that. Um, the reason that this research question is relevant is because biodiversity is important for having a healthy ecosystem and environment. With more species and more, yeah, with more species in a certain habitat, we can let more environments thrive and the ecosystem is more healthy overall. We wanted to recognize factors in a location that correlate to an increase of biodiversity, which is beneficial in keeping locations such as the San Joaquin Pond healthy. The San Joaquin marshes are more like a model for the natural environment that's outside, but we can use this as a model so we can find out different information about different areas, not just this Joaquin Marsh specifically. Here's some examples of biodiversity. When we went to the marshes, we took some pictures of some of the plants that were growing around the area. So yeah, NESTA can help us with factors. This can help us with environments that lack biodiversity in other areas. Our hypothesis that if the transect is further from the walking path, there will be a higher diversity index. So we believe that there will be more diversity. Yeah, we believe that the closer you are to the pond, then there will be a higher number of species present due to nutrients being in the area. and so there will be more opportunities for species to thrive and grow. Um, yeah, there will be different supply. It will supply nutrients for different plants and animals and overall increase biodiversity. The methodology of samples collected were that we use variables of vials of soil with eDNA collected from the marsh. They came from independent variables of experimental ponds 2, 3, 4, 5, 8, and 9, and transects ranging from A, which was closest to the path with an elevation close to 18 meters, to D, which is furthest from the path with an elevation close to 0 meters. The purpose of this samples collected were that they were analyzed according to the eDNA contents to determine the organisms that left behind those traces. And the eDNA contents would vary according to elevation, which the elevation was from 0 to 18 meters, and determine the proximity to that point. If there's more eDNA traces, that would mean there's more biodiversity, because the more eDNA, that means that there are more different types of species. Some of the metadata we recorded, it was regarded from location, so latitude, longitude, and altitude within the San Joaquin Marsh or Aldrich Park, and the transects we had were ranging from A to D. So yeah, our primary focus was the altitude, experimental pond number, and transects from the San Joaquin Marsh, and we also used the kit number, images, plant sample ID, and taking notes. Our prediction was that the increase in soil nutrients caused by closer proximity to the pond water will result in an increase in species diversity. The taxonomic groups being targeted were there were all the groups on the right, so those were the ones targeted for the metabarcoding. And metabarcoding is the barcoding of DNA or RNA or environmental DNA and environmental RNA in a manner that allows the simultaneous identification of many taxa within the same sample. The taxonomic groups that we're studying in particular are the phylum, which is the principal taxonomic category that ranks above class and below kingdom, and then genus, which is the taxonomic group that ranks above species and below family, and species, which is a group of living organisms consisting of similar individuals capable of exchanging genes or interbreeding. Some of the species in the dataset include the sea clubrush, soft brome, and scutch grass. This is mostly contained of PITs, which is a metabarcoding code for plants. And this is some diagrams we have of like pictures of the plants and the trees. So this is our graphs. So the reasoning for these graphs is that it shows that there wasn't really a difference between ponds 2, 4, and 5, which is why it says there's no clear trend. The x-axis is the altitude of samples taken, which is increasing, and the y-axis is the number of species listed. So there's no clear trend for the experimental pond 2, 4, and 5, and that's because it's just trying to show that that each pond doesn't really have a difference between each other. This is the comprehensive data graph of all the data that we had in general. So, yeah, like I said before, the x-axis is the altitude of sample taken, which is increasing, and the y-axis is the number of species listed. So that red line shows the downward trend. So there's a slight downward trend, which indicates a decrease in species presence as distance from the pond increases. And yeah, this shows variables, reliability, and eDNA presence. My interpretation of the findings was that, or my group's interpretation, was that the data shows a steady decline of number of species as the distance from the pond from where the data is taken increases. So this proves our point that diversity increases near bodies of water as the pond has higher nutrients to grow plants and other organisms that might live in the area. If you're further from the trail, you may also have be further from human interference, and human interference can decrease biodiversity by damaging or killing plants and animals. And animals will also inhabit areas near plants or other animals for food, which creates a positive feedback loop for biodiversity. Thank you so much for listening.
I thought you had really good like slide organization, and the data like analysis at the end was like that was really good. How you had the graphs and the actual data, and I thought it was really well put together.
I like how you had different graphs representing something different, and also everything was really well organized as well.
Yeah, I thought the slides were like really well organized. And I liked like all the like definitions and like the graphs too. Like everything was really easy to read and like flowed really well.
You.
I'm gonna start sharing my screen already. So,
I finished with comments, so we're good.
before I start my presentation, English isn't my first language, so if I butcher something, I'm so sorry. Okay, so hi, my name is Yexelen Perez. I am in the Tuesday 12 p.m. Class, and this is Team 15's presentation. So our research question or our prompt was, how does human activity in Aldrich Park and the San Joaquin Marsh affect plant population density and biodiversity? So we have here sample areas. So I'm not gonna say much because I don't want to bombard you with information. But for the San Joaquin Marsh, it is about 300 acres of land, coastal freshwater wetlands, part of conservation efforts of California wetlands and improving local water quality. There's about 200 and more animal species and 250 plant species. And so for Aldrich Park, it is about 19-acre park central to UCI campus. About 37,350 UCI employees are there, and the population of 20,420 as of 2023. And so the park is frequently managed and is often visited by dogs and squirrels and etc. So for the upcoming slides, I will not be saying all of them. I will just be summarizing it. So here we have sampling techniques slash data. So we decided to samples were collected using label vessels and were packed 75% to the brim with little to no rock chunks, and sample analyzed utilizing meta barcoding to visualize data in a metadata and taxonomy table. And so variety of plant species were observed and conditions of sampling data such as the weather, the GPS coordinates, plants IDs, and soil state. So if it's like dry or moist were also collected. And so variables of studies slash taxonomic groups of interest. So we have the plant population, density, and biodiversity, human activity slash population, and the PITS. So our hypothesis. So basically what it's saying here is that a higher human activity reduces plant density due to activities like mewing. Aldrich Park, with its larger population and more frequent human activity, will experience greater impact on plant density and biodiversity compared to the San Joaquin Marsh. So you see here, these are pictures here of the Aldrich Park, and at the bottom it is at the San Joaquin Marsh, one of the ponds. And so it says here, why is this important? So for rational, we said that we were interested in how plant abundance is affected by human activity in a populated, industrialized city. And so for relevance, here we have plant population density and biodiversity are crucial for our economic health and addressing climate change. The well-being of our environment depends on how human manages it and protects our biodiversity. Yeah. So our prediction overall, we said that a higher level of human activities will result in larger plant population density. Aldrich Park will also have a lower plant population density and biodiversity. And so necessary experiments. So we're saying that visiting the marsh or Aldrich Park bi-monthly to monitor vegetation and specific pond/pots, noting where they have been mewed and recording quantitative data such as people that are visiting it and over the years of how like stuff changes over time, basically, and track the areas with litter or pollution and test soil health and collect sampling data for the meta barcoding. And so the next slide. So, well, before I go to the next slide, here is a picture we took while we went to the San Joaquin Marsh to collect sampling data. And so our next slide here is basically saying that compare monthly plant species data to identify trends in species growth or decline in marsh/the park, track population changes, including new students in Aldrich Park that are visiting or that are employees at the marsh, compare population size between Aldrich Park and San Joaquin Marsh. And so the next slide is our predicted/finding and interpretations. So before I start reading, here's a graph, I guess a graph that's going down. At the bottom, it's the human activity, and on the y-axis, it's the plant population density. So what it's basically saying here is that human activities negatively impacted plant population density. As activities increases, plant density will decrease. This supports the hypothesis that humans' interference, such as mewing and pollutions, harm the plant population. And so the last slide is saying, this is another graph, and it's saying that based on our prediction findings show that Aldrich Park, with its larger population and higher human activity, has a lower plant density than the San Joaquin Marsh, validating the hypothesis that plant density decreases with increased human activities. And that's it. Thank you very much.
I liked your theme with it. I thought your theme was really nice and it was very cute and well organized. And overall, the information was very good, and I could tell that you had a thorough understanding of the information.
Thank you.
I really liked how like you had like a lot of information on your slides so that like we could read it, but also like I liked how you summarized it too. It made it like a lot easier to understand it. But if there's like a part I wanted to know more about, I could like read more about it.
Thank you.
That's what I thought too. Like, there was a lot of words, but like when you explained it, like you shortened it up and made it a lot easier to understand and got to the point.
Thank you. Yeah, I wasn't gonna read the whole thing. It was just too much for me.
Well, I think that's pretty much it, right? And then
Yeah.
I'll end the Zoom recording and then I'll download that. Or
Okay.
I think that's how
Yeah.
that works. I'll figure
Uh,
it out.
all right,
And then
thank
does
you.
anyone know how to upload like the slides to like the PDF